Hi, this is Sue Glenn. We are working on the chapter on energy and nutrient relations in the textbook Ecology, Concepts and Applications, and that's the 8th edition by Manuel Molles and Anna Sher. And we're going to spend a little bit of time figuring out how organisms gain energy and the proper nutrients. Organisms are classified based on how they get their energy, and there's a lot of different classification systems, but we call these feeding levels, they're trophic levels. And we're going to start off with the autotrophs, which are the ones that are able to use inorganic sources of energy and carbon in order to feed themselves. Auto means self, troph means feeding. A trophic level is the feeding level of an organism. It's where they get their energy from. It's a way of classifying organisms. If they're photosynthetic autotrophs, they use carbon dioxide as their carbon source and sunlight as their energy. Chemosynthetic autotrophs use inorganic molecules as a source of carbon and energy. And then in the next presentation after this one, heterotrophs we'll be talking about, which use organic molecules as both our sources of carbon and our source of energy. So I'm going to start off talking about chemotrophs, chemosynthetic autotrophs. And what they do is they synthesize organic molecules using carbon dioxide and inorganic molecules as energy sources as well. And they were first discovered in 1977 that there was a whole community based on these chemosynthetic autotrophs living at the bottom of the ocean. And when they were exploring the bottom of the ocean, they thought, well, there's not going to be much here other than detritivores, things that are eating dead things. And they didn't expect to find that such a vibrant community that was going to be based around basically these chemosynthetic autotrophs getting energy and putting it into the food web. The energy they were getting was from chemicals that were discharged from volcanic activity in the oceanic rifts, so the cracks in the ocean bottom. And the autotrophs are really dependent on chemosynthetic bacteria. There's chemosynthetic bacteria that are free-living forms that can live in hot springs, they can live in caves, they can live in lakes, they can live in these hydrothermal ocean vents, but there's also ones that are not free-living. They live within the tissue of invertebrates, so they're basically a symbiotic relationship where the invertebrate brings in what the bacteria needs, and the bacteria in return is going to provide oxygen and sugar to the invertebrate. So how do these ecosystems survive? Way down at the bottom of the ocean where there's no light, there's no photosynthesis. These chemosynthetic bacteria are basically living within the bodies of these giant tube worms. And these giant tube worms down here are like 4 meters long. These are huge. I thought a giant tube worm would be like a couple of feet in length. That would be a big worm. But these things are towering over. These are giant tube worms. This is a figure out of your book, and they're showing you how this ecosystem is structured. And just going to zoom in here, we can see that the nice cold seawater is mixing with this hot water coming out of these ocean vents. And so it's bringing down the temperature of the hot water to something that these organisms can live with. And that water contains hydrogen sulfide, which is coming out of the vents. So the giant tube worms, what they do is they bring in oxygen and hydrogen sulfide. They have hemoglobin in their system, so the top of the tube worm is actually red in color. Let's just move over in this figure. The bacteria are actually in the tissues of the tube worm, and they are chemoautotrophs, so they're able to take these hydrogen sulfide and oxidize it, go through a process of chemosynthesis, which is very similar to the process of photosynthesis, where they're mixing it with the oxygen, and they are actually getting the energy out, which they can use to build a sugar molecule. That sugar molecule is an organic source of carbon that can be then used by the giant tube worm in the process of respiration. I'd like you to pause after this slide. The next slide, there is a video from the BBC that is part of a series that was done by Dr. Ian Stewart on Earth, the Power of the Planet, about these great chemosynthetic bacteria. So I'd like you to take a moment and watch that video. So when we're talking about chemosynthetic autotrophs, we have been talking about these bacteria that are able to oxidize sulfur or hydrogen sulfide or thiosulfate in order to get their energy. There's other bacteria that are able to oxidize iron-based compounds, hydrogen-based compounds, carbon monoxide. So there's quite a variety of bacteria out there that are getting their energy from non-organic sources. And that makes it interesting when you are looking for life on other planets, because you don't necessarily need something that's based on photosynthesis where you're a certain distance from a star. You could actually be quite more distant from the star and be using the energy from these compounds being released from the center of your planet or your moon, and you're using those to actually form the base of an ecosystem. So instead of just looking for Earth-like planets, you could be looking at the moons of Neptune, and beneath the ice on their surface, there might be things happening in the liquid underneath that are producing life-based ecosystems or are based on something other than the energy from the sun. In addition, there are nitroviolent bacteria that get their energy out of nitrogen-based compounds, and they are super important to nutrient cycling of nitrogen. We need nitrogen, obviously, to make our enzymes because they're protein-based, and so nitrogen is something we need in our diet. The plants need nitrogen for their enzymes. Everybody needs nitrogen, and so these bacteria are really important. They can take ammonia out of the urine that's been released from the animals. They can oxidize that to nitrite, and then there's different bacteria that oxidize the nitrite to nitrate, and then the plants can take back up that and use it to produce their proteins. So you can see here the bacteria are in the soil. Sometimes they actually live within the roots of the plant, and they're able to convert the ammonia into a more useful compound, but the actual bacteria get their energy out of this, and they're able to use that energy in order to grow and reproduce. Now, when we look at light-based ecosystems, we're going to be dealing with the energy coming from the sun, which means that you have to be aware of the electromagnetic spectrum. There's different wavelengths of light. Light is basically a photon that is going through space at a wave, and the properties of the wave, the wavelength, is an indication of how much energy is in that light. So if we're looking at low energy radiation coming off the sun, that's things like radio waves and microwaves, and they have much longer wavelengths. And then we have higher energy in the visible light. The visible light, as you know, breaks off into the spectrum of colors, from the lower wavelengths being the red up to the faster wavelengths being the violet. And then even faster wavelengths than that are ultraviolet, faster X-rays. This is such high energy, it can go right through your body, just get stuck with the bones. And gamma rays can go right through the body, go right through the bones. And those ones that are longer wavelengths than visible light, they're much more dangerous, because that high energy light is able to shake up our DNA and break it, and we try to fix it, and we don't fix it right, and it's still working, but it's broken, causes mutations, causes cancer, it causes damage to biological molecules. So we have a solar powered biosphere where light is the base of that biosphere. Photon is a particle of light with energy. So infrared light is your longer wavelength light with the lower energy. what infrared will do is it'll cause the molecules to move faster. You think about heat, the molecules are moving faster in infrared light, and so that's longer wavelengths than visible light, and increases the motion, the movement of the molecules. Ultraviolet light, shorter wavelengths of light, so that's higher energy. So these are shorter wavelengths than the blue and violet in our visual spectrum, and that can destroy biological machinery. What we're really interested in the biosphere is what wavelengths of light can plants use, and that is the photosynthetically active radiation, which so we just use the abbreviation PAR, and we measure that as the amount of photons within those particular wavelengths. So photosynthetically active radiation is in that sweet spot in the middle. When we look at the energy coming in from the sun, it has to pass through the atmosphere before it gets down to where the plants are going to be using it. And so we're looking here at the spectrum of wavelengths coming from the sun. And on this particular diagram, we can see that the shorter wavelengths, so the higher energy light, are on the left side of the diagram, and the longer wavelengths, the lower energy, is on the right side of the diagram, which is the opposite of how we had it in the previous slide. And we can see that a lot of this very dangerous high energy light is actually getting absorbed so that it doesn't really get past the stratosphere down into the levels where we're going to find life, which is good because that's going to hurt us. And you can see we get a small amount of ultraviolet light getting through, which is the dangerous part, which is why you don't want to go out without protection. Then we can see that the visible light range gets through the atmosphere fine, and then we can find that the infrared and longer wavelengths are getting through depending on the wavelength at different levels. So this is actually from an astronomy site where you can see that we can look at the stars from the surface of the Earth if we are looking around visible light and a little bit into the infrared, a little bit into the ultraviolet. But then we have radio telescopes that are also able to see the radio waves and microwaves, so the longer wavelengths coming into the surface. The photosynthetically active radiation then are the wavelengths that are between 400 and 700 nanometers that have the energy that can drive the light-dependent reactions of photosynthesis. So it's not enough energy to destroy the organic molecules. And so photosynthetic radiation makes up about 42% of the energy in the sunlight that actually reaches sea level. And the pigments involved in photosynthesis, there's different pigments and they absorb different parts of that wavelength. But the energy actually available for photosynthesis then is only about 26% of the total. Here we can see the tree canopy is going to be blocking a lot of the light. It is actually absorbing that light and using that for photosynthesis. And you can tell that the canopy is green in color, which means the green wavelengths are not very tasty for the plants. And those are the wavelengths of lights that are getting through. This is looking at a boreal forest, so a northern forest, and we've got a canopy that's mainly a very dense canopy of coniferous trees, conifers, evergreens that keep their leaves, their needle leaves all year long. The canopy absorbs about 79% of the photosynthetically active radiation coming in from the sun to the surface, and it is reflecting about 10% of the light off of the surface. And then, so most of it is taken up by the needles in this canopy layer. The plants that are trying to grow beneath that in the darker area, they absorb about 7% of the light. The vegetation down near the forest floor is getting about 2%, and then actually reaching the forest floor is only 2%. So you can see that the competition for the trees to get their needles up into that canopy layer has got to be intense because they want to be able to get as much light as possible. That's their energy source. So we measure power as photon flux density. Photon flux density are the number of actual photons striking a square meter surface each second. The chlorophyll is absorbing the light as photons and getting the energy from that light, putting it into an electron that's ripped off of water, and using that to move that energy into energy carriers, that electron into energy carriers, so that we can, so the plant can put that energy into a sugar. As far as how much light you're getting on the plant, what your power available to you is going to then depend on if another plant's shading you, if you're on the shady side of a mountain, so the landscape's going to make a difference. The amount of water that is in the air, whether if there's condensation and clouds blocking your sunlight. So there's a lot of different factors in the environment that is going to affect the amount of sunlight that you're getting, and the amount of photosynthetically active sunlight that you're getting, and the quality of that light. Back in biology class, you learned about the different photosynthetic pathways. In ecology, it's important to understand these pathways because the different pathways have different advantages and disadvantages in different environments. C3 photosynthesis is the most common form of photosynthesis. It's used by most plants and algae. It was the first to evolve. And it is called C3 photosynthesis because when the plant first takes in the carbon dioxide, remember it comes in the little stomata, the little holes in the leaves, the carbon dioxide is grabbed by an enzyme called RUBP, ribulose bisphosphate, which is a 5-carbon sugar. That 5-carbon sugar now has 6 carbons because of the carbon dioxide, forming a 6-carbon molecule. That 6-carbon molecule immediately breaks apart into 2 3-carbon acids. So there's going to be 2 of these, and that's the phosphoglyceric acid, which is called PGA for short. You probably remember all of this. If you don't remember your general biology, you may want to go and review that and look up the C3 photosynthetic pathway. In order to get this carbon dioxide into the plant for photosynthesis, it has to open the stomata on the underside. Most of them are on the underside of the leaf, and that reduces the amount of water loss, because when you do open it up, you can have water escaping depending on the differences between the water in the leaf and the water in the air, and that was in our previous chapter on water relations. So in order to do photosynthesis, you are going to be losing water, and that water escaping the leaves is part of that pump that's going to be drawing more water back up from the roots. So it's one of the forces to keep the water moving throughout the plant as well. When we look at C3 photosynthesis, here's an example using a sunflower, and we've got a close-up on a cross-section of a leaf. When you look at a cross-section of the leaf, we have a large number of cells packed into the top of the leaf. Those are the mesophyll cells or the palisade parenchyma cells, and that's where most of your light's coming in, so they're going to be capturing those photons in their chloroplast for doing photosynthesis. But we have a looser array of cells down here at the bottom. These are called the spongy mesophyll cells because there's air spaces between them. So when the CO2 comes into the cell through the stoma, can go into those little air spaces, and that CO2 is picked up by the RUBP, makes 2 of these PGAs, and then it can go through this process 6 times, and it can make 1 sugar molecule, which is C6H12O6, your glucose molecule. C4 photosynthesis, very similar process. What it's able to do with C4 photosynthesis, when it first grabs that carbon dioxide in those air spaces, it is going to use a different molecule to do that. And the reason for using a different molecule is that this molecule is stickier. It has a higher affinity for carbon dioxide. So it'll work better when your carbon dioxide is starting to drop. And when are you going to have this carbon dioxide starting to drop? Well, it's when you start to lose so much water that your stomata close. Remember, the stomata have little guard cells that regulate whether the stomata are going to be open or closed. And if it starts to lose water, those guard cells will close the stomata. And then you can reduce your carbon dioxide that's available in those air spaces. So when it's able to work in those conditions, then it means it can actually take that carbon dioxide and move it further into the leaf and pull out more carbon dioxide that way. So it increases the rate of diffusion of carbon dioxide into the leaf as well. So you need fewer stomata to be open, and you can still do carbon fixation when they do close, when you're starting to dry out. So it is a water conservation mechanism in C4 photosynthesis, which means your plants will do better in a dry environment than plants that are doing C3 photosynthesis, and because you're able to live with less water loss. So what it will do, it'll take that carbon dioxide, it produces an acid, and that acid moves into cells that are around the veins, the xylem and phloem, and in the center of the leaf, and then it can release that carbon dioxide again there, and then when it releases it, it just goes through regular photosynthesis from that point on. This is a C4 plant, a corn plant. A lot of C4 grasses exist. They live in a drier environment, and so the evolution of this type of photosynthesis is predominant in the grass family. So corn is a very large grass plant, and when we look at when the carbon dioxide comes in to the leaf of the plant from the stomata, that carbon dioxide is picked up by one of these spongy mesophyll cells, which are these guys right here. So the carbon dioxide comes in, it is picked up by the enzyme PEP carboxylase, which is very sticky. It sticks to it so that the carbon dioxide can be picked up even if you have a very low concentration of carbon dioxide. It produces this C4 acid, which is where the C4 name for C4 photosynthesis comes from. Then it transports that from that mesophyll cell here up into a bundle sheath cell, which is going to be a cell that is around the veins. And the veins, remember, xylem and phloem from your general biology class. The xylem brings the water up to the leaf, and the phloem takes the nutrients away from the leaf. So that C4 acid has moved into that bundle sheath cell, and then the C4 acid will just release its carbon dioxide into the cell. And from this point on, it's just the same as C3 photosynthesis. So it just goes through the regular process of C3 photosynthesis. When it releases that, it produces the pyruvate. So the pyruvate goes right back here, picks up another carbon dioxide molecule. So you use this over and over again and transporting the carbon dioxide from the spaces in the mesophyll cells to the bundle sheath cells. So this works well in very dry environments because you can not have as many stomata open. You're not going to be losing as much water. The third type of photosynthesis is the crassulacean acid metabolism, which is much easier just to say CAM photosynthesis. It is very limited in what kind of plants can do this. So this is succulent plants that live in arid and semi-arid environments. Arid meaning dry, so they're going to be like deserts and semi-desert environments. You're going to find these plants. And the clever thing that they do is they only open their stomata at night. So carbon fixation will only take place during the night. So remember, in the nighttime, we know that the temperature is lower. So we have a lower temperature. So when you have a lower temperature of the air molecules are not pushing each other apart as much. They're closer together. So you have higher air density. So there is less space in the water for the water to go in between the air molecules. So your air molecules are close together. So the amount of space for water molecules to squeeze in there is going to be limited. Because of that, your saturation water vapor pressure is going to be much lower. It cannot hold as much. So when the water does, when the air condenses at night like this, it can't hold much more water. So your relative humidity goes up. So you have increased relative humidity. So the air is more humid, more saturated with water. So you're going to lose less water from your leaves. You're going to have reduced transpiration. That is going to reduce your water loss from the leaves. When you, or the stems, they don't usually have very many leaves on these plants either. They usually just have green stems for doing the photosynthesis. When you have closed your stomata, then during the daytime you can do photosynthesis, but you only have as much carbon dioxide as you are able to take in during the day, during the night. So once the nighttime comes, the CAM plants are able to open their stomata to take in the carbon dioxide at night. So this is a little bit confusing because it looks like the same diagram for C4 photosynthesis, but one thing they did wrong here was they make it look like this is going on simultaneously, but one side of the diagram is actually nighttime and one side of the diagram is actually daytime. So over here we have the nighttime and on the right side we have the daytime. So it looks like it's transporting from one type of cell to the other type of cell. Remember in C4 we were going from the mesophyll to the mesophyll cell to the bundle sheath cell, whereas in this one we're still, both times we are in the mesophyll cells. So this really needs to be like a solid line, daytime and nighttime. So at night the stomata open, they let in the carbon dioxide, it gets picked up by the PEP, making the C4 acid, and then it'll just wait with that acid. So it can have the acid sitting in the cells waiting for the daytime. When the daytime comes, the stomata are going to close so that they're not going to lose water. So during the daytime, remember the temperature is going to go up, which means your relative humidity is going to go down as the spaces between the air molecules open up to allow more water in. Your saturation, the amount that the air can hold, goes up so that there's more space for the water to escape. So you close up your stomata during the daytime. During the daytime, then that C4 acid will release its carbon dioxide and you go through the regular C3 photosynthesis. So C3 photosynthesis, C4 photosynthesis and CAM photosynthesis are really adding on an extra step. For C4, it's a step moving from one type of cell to another type of cell. With CAM, it's changing it up during the time of day. So really, during the day, you're limited to making enough sugar depending on how much carbon dioxide you were able to store the night before. Once you run out, you can't do any more photosynthesis. So because of this, you're going to grow very slowly. Your cactus is going to grow very slowly because it's just not able, doesn't have the machinery to store more than a certain amount of carbon dioxide. It doesn't have a little million leaves sticking out all over the place because they would be losing too much water and heating the plant up too much during the day. So because of this, these plants are going to grow very slowly, but that doesn't matter if you're the only guys around. So if you're the only ones that can grow there, then that's okay. If you grow slowly, that's fine. Nobody's going to shade you out. So when we look at the different photosynthetic pathways, C3 plants lose 380 to 900 grams of water for every gram dry weight of tissue that they're producing, which is a lot of water. The C4 plants lose a lot less, only somewhere between 250 to 350 grams. So the C4 plants obviously have an advantage in doing photosynthesis where you have less water available. And this could even be seasonally. You might have a seasonal drought during the middle of the growing season, and the C4 plants would have an advantage at that time. And the CAM plants, they have the biggest advantage in a dry environment, which where they only lose 50 grams of water for every gram of tissue that is produced. So the CAM plants do the best in dry environments. There was a question about photosynthetic pathways on the SAT a couple of years ago, and then students who are not allowed to share their SAT exam questions did anyway, and produced a few memes. So I've just added in some of the little memes that they had on photosynthetic pathways. At the end of the chapter section 7.1, there are concept review questions. You should be able to answer these. Number one, what environmental conditions favor plants with C3 photosynthesis and why? So when do those plants have an advantage? How are C4 and CAM plant photosynthesis processes similar, and how are they different? And the third one, why does the ongoing increase in atmospheric carbon dioxide not give guaranteed advantage of C3 plants over C4 plants? So with climate change, we are increasing the amount of CO2 in the air, which is a greenhouse gas. It actually holds more heat in the air. With that increased atmospheric carbon dioxide, why doesn't it give a guaranteed advantage to C3 plants over C4 plants? So you have to think about carbon dioxide and water loss to answer that question. Time for a break? If not, you can move on to the next presentation. We're going to be talking about heterotrophs and how they get what they need. See you then.